Hi, this is Miss Litton, and this is a Friday afternoon, and this is my wonderful 7th period. Say hi.
Hi.
All right, and we're starting in, well, you've already started reviewing. This is just drawing attention to a few things because you've been reviewing. So the first thing I want to talk to you is a reminder that we did this class as units to break it down into individual units, but that's not how the AP exam approaches it. You know that because you've already had one AP exam. You need to know the 4 big ideas. The 4 big ideas then have what's called enduring understandings. And then those enduring understandings, this one enduring understanding, change in the genetic makeup of a population over time is evolution. Okay. So that's what evolution is, a change in the genetic makeup. Then there are 4 essential knowledge ones that support that particular enduring understanding. Natural selection is a major mechanism of evolution, so we need to understand the process of natural selection and how it is a mechanism for evolution, right? Now, there are 55 essential knowledge pieces, okay? So I don't know if you can see the smaller picture over here on the left. Here are the 4 big ideas. Those 4 big ideas are supported by these enduring understandings. Within these enduring understandings, you have this essential knowledge accompanied with the science practices, okay? So, and then those give you learning objectives. If you look to see, remember when I would go LO 3.5? That's putting together both science practices and essential knowledge. So at the very basic, make sure you're familiar with the 4 big ideas. I am in a review. What I have, Big idea 1 presentation, big idea 2 presentation, big idea 3, big idea 4. If we can get through all of them, I think this is like 105 pages, just big idea 1. If we can get through 50 pages today, quickly reviewing and addressing, then we'll get half of big idea 1 done. Next time we'll do the other half of big idea 1. Do you see what I'm saying? So we can get through all of them. I want to remind you what the science practices are, okay? If we look at the science practices, the student can use representations and models to communicate scientific phenomena and solve scientific problems. The students can use mathematics appropriately. Oh, well, we have a 2-page formula sheet to see if we can use mathematics appropriately. The student can engage in scientific questioning to extend thinking or to guide investigations within the context of the AP course. The student can plan and implement data collection strategies. And they ask you questions like that in the multiple choice. What are they showing here? What data would be the best data to collect? What should they have known? How should they set up this experiment? Appropriate for a particular scientific question. The student can perform data analysis and evaluation of evidence. So remember, claim, evidence, reason, that's part of that. The student can work with scientific explanation and theories. The student is able to connect and relate knowledge across various scales, concepts, and representations in and across all domains. Okay, so those are your science practices. Where did we learn about those science practices? We did them within our labs and our investigations. So we are not tied to any particular labs other than the fact that we need to do two labs per each big idea. So, and that is what we've done. I think we did 1 bonus lab, but you need to have 2 labs per each big idea. So you want to be familiar with those and the content that it reviews, as well as what you learned how to do in that lab. How did we measure photosynthesis? How did we measure enzyme activity? How did we measure transpiration? How did we know we could manipulate DNA and change an organism's traits, right? We're doing that right now. So you want to make sure you pull that in. As a side note, I just thought I'd remind you of what the exam is currently. You are going to be the last group. They did update the test in 2013. They're going to have another update in 2020. Yeah, yay for you. For you, your multiple choice, you have 69 questions. That's what you do first, and you've done one practice with me already. An hour and 30 minutes, it's 50% of your exam score, and you have 6 grid-ins. Starting next year, they have no grid-ins, the same amount of time, and only 60 questions.
What?
I know. And
less questions on the...
Yeah.
Cool.
Free response.
What is that?
Free response. Oh, I don't, skip. Remind me later. Maybe they're going to be harder. I don't know. But you are going to be the last ones who have 8 questions. From here on out, they're only going to have 6 questions. So take pride. Take pride, my friend. Maybe because they know they're going to transition, maybe it'll be a slightly easier test this year. I don't know. So I just wanted you to know you're the last ones to go about it that way. Okay? So now I'm going to go back. I have given you since day 1. Do you see this document right here? I gave you this document. It's in your review folder underneath learning objectives, or I can't remember exactly what it is, but if you scroll down, you have this. There we go. Go, scroll. You have this document, okay? And this was done by a man named Jeremy Kahn for Clear Biology. I honestly don't know how I have it, but I do have it. But it's in your folder. And so this was an assignment where he would have them identify how well they knew it. But I like it because it's one of the easiest documents, which shows the enduring understanding and then the essential knowledge. I have also provided all the learning objectives for you. Do you remember how I told you it's the 4 big ideas with the enduring understandings that are right here, and then they put in the science practices. I have included just all the learning objectives. That would be another excellent review document. It just go, can I do that? Can I do that? Can I do that? Because that puts together this with the labs. Those are all, if you have a short amount of time, focus on this and those learning objectives. Work through them and see if you can do those. I have told you since day 1, every time I've identified every single learning objective on all your notes, so you would know that. Okay, but we can't do anything about that now. We've reminded. But what we're going to do is we're going to work through these, but you could print this off, and everyone you go, okay, I know that, you could highlight it. Today, I'm going to talk about Big Idea 1, and in Big Idea 1, there are 1, 2, 3, 4 enduring understandings. If we can get through the first 2 enduring understandings, then we will also have covered 1, 2, 3, 4, 5, 6, right, essential knowledge topics. Okay, do you see where we're going and what we're doing and why we're doing it? You okay with that? All right, so now let's go in here and let's do the first big idea number 1. The process of evolution drives the diversity and unity of life. So that is our first big idea. It's all about evolution. It drives the diversity and the unity of life. So when I look at this picture, when you think, how does this demonstrate evolution? What's a word that comes to mind when you see this picture? How does it demonstrate evolution? Any ideas there? Are they all the same beetle? There are what? Variations. Why are there variations? You could talk about why were variations created? Why are those variations persisting? How could the variations have come about? Yes. Mutations could have brought them out, right? And then why are they persisting?
Natural selection.
Yeah, natural selection is selecting the best fit beetle for that particular environment, right? So I need you to start seeing big ticket things. You're not by units. You're pulling everything together you've done so far. And you're thinking, what adaptations do these beetles have for their particular environment where they are living? Okay? On evolution, we need to make sure we remember the difference between macro and microevolution. Microevolution is within a species, but it could lead to macroevolution, which would be a whole new species, right? Okay, based on that, let's do enduring understanding 1A. This is underneath this big idea. Change in the genetic makeup of a population over time is evolution. If you're taking notes, I wouldn't bother typing all that out. I would write big idea 1, and then underneath that I'd put EU1A, because you have a list of them. I would just print them out, right, to look at. So change in the genetic makeup of a population over time is evolution. We've talked about that. But first thing is natural selection. More offspring are made than can survive, right? Of those, there will be variations amongst those offspring. Those variations, the only ones that are important are the ones that are in the what? DNA. Not because one got more nutrients when they were developing than another, right? Not because one got kicked in the head and one didn't, okay? We're talking about you have it in your genes so you can pass it on to your offspring. Some of those variations are going to be more adaptive than other variations, and those are the variations that are going to get passed on because they have the greatest fitness, right? That's the big ticket here about natural selection. So variations are a prerequisite for natural selection. Natural selection does not work unless there are variations, okay? Then you're rating those variations, right? What makes you the most fit? So we looked at population genetics as a way to measure quantitatively variations within a population, right? Because variations are important for that. So we looked at the alleles, and we said for diploid organisms, they have 2 copies of every allele, yes? And if we simplify it and just say there's a dominant and a recessive allele, we all know in here that genetics is not always that simple, right? But we're just saying, for simplicity's sake, you have a dominant and a recessive allele. And for us, what we did is we said that the dominant allele in our equations was equal to what? What was the letter we used? P. And the recessive allele was equal to Q. And what we said is we said P plus Q equals 1. We said however many alleles, in this case the P's would be all the B's, we add up all the big B's, we add up all the little B's, together that'll give you 100% of the B's, right? Okay, so if P is 0.8, if 80% of the alleles are big B, that means only what? 20% are little b. That's in your gene pool. How it plays out, how you pull them out of the gene pool, you could have 2 big B's, right? You could have a big B and a little b, or a little b and a big B. That's why we have 2PQ, right? Or you could have 2 Q's, right? And all we have to do is say, okay, well, if big B is 0.8, if P is 0.8, what are the odds that from this gene pool of this population that I am going to have an individual who is homozygous dominant? 64%, right? Because big B is 0.8, right? And another big B is 0.8. Together, multiplying together, 0.8 times 0.8, right? Right? So now we would say that there was a 64% chance that you would come out homozygous dominant. What are the odds you would come out homozygous recessive? Well, if big B is 0.8, then little b is what? 0.2. That's your Q, right? So 0.2 times 0.2 is 0.04, so 4% would be homozygous recessive, yes? And then what are the odds of being a heterozygote? Well, you could be PQ or QP, so that's why we say 2PQ, yes? Right. So 0.8 times 0.2 is? 32%. 0.8 times 0.2 is? 16. Times 2 is 32, okay? So that, what you're talking about right there, is Hardy-Weinberg, okay? And we said, if you want it to stay the same, 0.8 and 0.2, if you want to maintain that and have no variation between 0.8 and 0.2, it's going to stay that way, you have conditions you have to meet. If you fail to meet those conditions, then what's going to happen? The allele frequency changes, and therefore what occurs? Evolution. And basically what we said is, it's very difficult to meet those 5 conditions, so chances are evolution is going to occur. So these are our 2 equations you want to be familiar with, okay? Basically, these will stay continuous only if you meet the 5 conditions. Otherwise, things are going to change. You could have small incremental changes, right? Microevolution that leads to a macro, a larger one, right? Okay, to a whole new species. So, can I come back to this? Okay. All right. So, what are the agents that cause change in gene frequency? So we know mutate. Remember the 5 fingers?
Yeah.
Yeah. So let's do the 5 fingers before we even look at this. All right. So this one says what? Small populations cause change. Non-random mating, that's the ring finger. Sorry, we said pinky is a small finger, so A pinky leads to small populations are more susceptible to genetic drift. Do you remember that? And then we said the ring finger is, hello, non-random mating. And then the middle finger is mutate. Look, he's giving that to us. Sorry, not really. We're recording this for all the world. So the middle finger, we remember mutations. Pointer finger is, I'll see you over there. Are you going there?
I'm taking one for the team right now.
All right, thank you. Pointer finger is what?
Natural. No.
Gene flow.
Gene flow.
Yeah, gene flow, right? Gene flow is new alleles coming in or alleles leaving, right? And then what's the thumb?
Natural selection.
Natural selection, because the thumb is very adaptive. Okay, so this is the middle finger right here, mutations. Gene flow causes change. Non-random mating. Okay, random mating is she doesn't care which lion is behind her. Okay, that's random mating. But she does care which lion is behind her because she wants her offspring to be strong. Now, if this, do you remember the two things we talked about in sexual selection? What were the two hypotheses about why she chose this particular lion? Runaway and good gene. Chances are this is more along the lines of good gene, right? It's not that lions are evaluated like birds are on how pretty they are. So probably she says, this guy has the best genes, I'm selecting him. Or he just commandeered all the females more than likely because he is the biggest, strongest, and has good genes, and so he has sex with them. Okay. Now, your next one, okay, is genetic drift. That's due to small populations, right? So it may not be that the green frogs are very adaptive, but it could be that some random event took out a bunch of the variation, so only green is left. That is more likely to happen if you have a small population than if you have a large population. Exactly. Okay, and another thing that can happen to genetic drift is founder's effect to represent genetic drift. And that is if you have a large population and you just pull out a small percentage of it, that's not necessarily representing the whole. So the new population that gets started maybe it has some mutations that the other ones don't have. Okay. Next, natural selection. That's your thumb. So nature is selecting who is best adapted. If you're a giraffe, it is preferential that you have a what?
Long.
Yeah, but not so long that if you lean over, your blood pressure causes blood to squirt out your eyes, right? You follow me on that? It could be too long. It's nice to have sharp teeth, but not so sharp that you're constantly cutting and bleeding your mouth, and so you end up, right? So it's not, you don't just keep going and keep going to one extreme. So artificial selection happens, naturally in our environment where we say this bird is better than this bird, this wheat crop is better than this wheat crop. We're selecting for those particular traits. And Darwin looked at that. We can see that in dogs. That's artificial selection. When you see something gaining like more and more oil in the corn kernel, the trees are taller and taller and taller, that's directional selection. Can directional selection lead to speciation?
Yes.
Yes, you're favoring one extreme. Okay, you're favoring one extreme. Then there is stabilizing selection. For instance, if you give birth to a baby, you don't want it to be too big, but you don't want it to be too small, so that's stabilizing. You don't want to have too many eggs, you don't want to have too few. Does that lead to speciation? No. It favors the status quo. It favors the norm. Okay? Then the next one was disruptive selection. Does this lead to speciation?
Yes.
Yes. Okay? It's either better to be a really dark snail and be on this part of the leaf, or a really light snail and be on this side of the leaf. So you're favoring both extremes, which can lead to two different species. So how do you get these variations where you would have a dark snail and a light snail? Well, mutations can cause those variations. So you could have a visitor bringing in new traits, or you could have some sort of disruptive selection, like for Darwin's birds, they were finches trying to decrease what? Competition. So some favored larger seeds and some favored smaller seeds. That would be disruptive. So all of these things are promoting variations. When you have mass extinctions, you could have adaptive radiation spreading out to new areas. Okay, when we talked about Hardy-Weinberg and Diploid, notice it doesn't matter if little a is 0.9 or 0.1. If little a is 0.9, that means big A is 0.1. And if little a is 0.1, that means big A is 0.9. But notice that the heterozygote stays the same number, right? Because one is reflective of the other number, right? So you can't necessarily eliminate one or the other because you can always carry it in the heterozygote, unless it is an autosomal dominant lethal trait, and that's going to be probably eliminated, right? Because if you just have one of them and that kills you, that would tend to rule that trait out. Okay, next, if we're saying these types of things lead to evolution, we're saying that mathematically we can figure that out, right? What evidence do we have of evolution? And I gave you, and that would be common descent. So what's this one about? Fossils. And we said to be a fossil, you have to be about what? 10,000 years old. What's this one? Biochemical, right? So we can take a look and compare similarities between organisms and what they have, the enzymes that they're using, and we could maybe point back to that common ancestor right down here who is what? Luca, right? So similarities in DNA and proteins. What's this one? Yeah, biogeographical, right? So we can see when the continents were together, then you have, let's say, uniformity of a fern plant. But when the continents separated, and about 65 million years ago when the mammals were evolving, you started to see unique traits come up. They're solving and living in similar environments, like you're an herbivore and I'm an herbivore, but we're on two totally different continents and we exploited it. We had different mutations, different selection pressures, and so we both are playing that same role in the food web, but we might look markedly different, okay? What's this one about? Anatomical evidence. Anatomical. So on anatomical, we had several things that we looked at there. Okay, one of those, which is demonstrated by this diagram right here, what is this showing you?
Convergent.
No, not convergent. Well, I guess you could say none of them are actually convergent evolution on there. What is it? Not analogous, homologous. Homologous structures. Homo means same. Homologous structures, humerus, radius, ulna, carpals, metacarpals, phalanges. It does not matter if you're a human, a cat, a whale, or a bat. Same bone structure, okay? They might have longer phalanges or a longer or shorter humerus, but if you have the same anatomy, then you probably have the same DNA and ancestors coding for those traits. I'm using mine to pick up my phone, but somebody else might be using theirs to, yeah, or fly. Was that swimming?
I thought you were.
Okay, I'm sorry. All right, I don't know. Okay. Anyway, exactly. Okay, now, anatomical homologous structures was one. What was another one? What? Developmental. We could look to see and compare how organisms developed and if they have the same. Remember we learned when a happens all the time. When a sperm fertilizes an egg, it forms a diploid, what? Zygote. It undergoes what? Mitosis, right? And forms a solid ball of cells called a morula, and then a blastula and a gastrula, right? And we can look at similarities in development and go, okay, you must have had a common ancestor. What's another one?
The outdated structures.
Vestigial structures, yes. Vestigial structures point to a common ancestor. If you can move your ears, maybe in your ancestral past, you were moving your ears. But it's not necessarily a requirement now. I would more point to something like a salamander that's blind in a cave, right? But he has vestigial eyes. So that tells you at some point he probably did have eyes. Or a penguin who has wings, but he doesn't what? Fly with them. Maybe uses them for something else. What don't we give as evidence for anatomical structures? What? Yeah, did I just say it out loud? Yeah, it's analogous structures. When you have the same function. And that is a result of what kind of evolution? I've heard it. Convergent evolution. I am swimming in the water and you are swimming in the water, so it's adapted to have fins, right? So it doesn't mean that our fin structure is the same. It just means we have fins in the water, right? And so we're responding to our environment in the same way. What is this one right here?
That's like those genes.
Dating. Yeah, so that, that's okay. So that's your geologic timescale, and we're looking back in time and measuring mutation rates, right? We're looking back in the fossil record and what we would predict for evolution, starting with unicellular organisms, then multicellular organisms, simpler plant like mosses, and then working our way up to gymnosperms and then angiosperms. You can see that over time, and it's kind of looking at fossil record and maybe even potentially biochemical, right? And looking for those similarities. All right, so that was the big ticket overview. So fossil record, we've already mentioned about fossils. I wanted to point out there are transitional fossils once again, because we would say from a fish would evolve amphibians, from amphibians you would evolve reptiles, and from reptiles you would evolve mammals, right? From therapsids. All right, so there are transitional fossils. You can look at the evolutionary history of the horse. What kind of selection does this look like to you? Directional selection, right? As their environment changed, as they lived from a forested environment, and as the continents were moving, and as a result it changed from a forested, more watered area near the equator to a drier area and into a grassland area, then it was better to be larger and bigger so you could run away faster and be above the grasses. So that would be a good example of directional selection. Biogeographical, we already talked about this. We already reviewed it. I wanted to see if you could come up with it without me showing it to you. Here's some other biogeographical evidence. Australian marsupials. So on this case, what I would say is you would see there was probably some sort of common ancestor, so this might be more along the lines of parallel evolution, right? Parallel evolution, I'm saying somewhere in our past we had a recent ancestor, or we had an ancestor, and so both of us are small herbivores, small furry herbivores. And so you can look at some similarities between what we have here in the United States and what you might find in Australia. Homologous structures, we covered that. Versus analogous structures, we discussed vestigial structures and embryological developmental structures. And the biggest one today to look at, if you have access to proteins or DNA or some sort of enzymes, you can look at biochemical evidence. I'm gonna skip that because we got done with the one enduring understanding. Okay, let's do another one. What time do we have? 2:59, 6 minutes. Can we finish another enduring understanding?
No, we can try.
Let's try. Organisms are linked by lines of descent from common ancestry. Okay. Look, the kingdoms. Here we know this. Living things are organized, made of cells. They acquire materials and energy. They maintain homeostasis. They respond to stimuli and reproduce. They grow and develop. They adapt and die. Yay. Instructions for life is located in your DNA. You know the genetic code, and this is universal. Okay, the genetic code is located on the mRNA. Every 3 bases on the mRNA is called a codon. Okay, who determined that a particular codon should be UUG? If this is on the mRNA, the DNA must have been AAC. Right? There are no T's, right, in the RNA instead of U's, okay? So the DNA determined the mRNA codon in those DNA that are actually translated. And then from the codon, you can determine what amino acids, because every 3 bases codes for a particular amino acid. Right? So this kind of showing you DNA. Here's the DNA strand. Transcription gives you the mRNA. Every 3 bases is called a codon, which codes for the amino acid. So ultimately, the protein is determined by the DNA. Now we know there are all different kinds of controls in there, right? Chromatin, what DNA is available, epigenetic inheritance, right? We can start to pull that all together. We could talk about whether RNA polymerase can bind to the promoter, if it is inhibited from binding to a promoter because there's a repressor, or if it's not helped because there's not a transcription factor. Then we can talk about post-transcriptional control. Remember, introns and exons are actually expressed and the introns are cut out. Remember that? The speed at which it can leave the nucleus, if it can bind to a ribosome without being degraded, and the protein once made has to be folded, right? We can remember all of those things. I want to remind you that the DNA is not just found in the nucleus with this coding information, but it's found in mitochondria and where else? We don't have them, but chloroplasts. And that shows us that mitochondria and chloroplasts could have been free-living prokaryotes, right? Who, because they have their own DNA, and the DNA is circular like bacteria, and they have ribosomes similar to bacteria, and they're self-replicating, and they're double membranes. Remember that? That was evidence for the endo-symbiotic hypothesis. Good. Okay? Glycolysis, who does it? All cells. Does it require oxygen to do glycolysis? No, it does not. And you're ultimately taking glucose and breaking it down in how many steps? 10 steps, 10 enzymes, and you end up with 2 pyruvic acid. To get the party started, you have to invest how many ATP? 2, but you get back 4, so you net out 2. If you're small and unicellular, you're fine with that, okay? I got my 2 ATP. I'm golden pony boy. But you need to be able to regenerate in order to get the 2 ATP. Remember, you have to reduce some NAD and make some ATP. If all your NAD ends up getting reduced, you're going to be in trouble. So we need to develop a way to oxidize it so we can do glycolysis again. What is that process called? Fermentation. You have 2 options on the fermentation, right? But because of the universality of that, yeah, then we can say probably there's some common ancestry there. We even have some of the same enzymes as that. Okay, prokaryotic versus eukaryotic cell, we already talked about. This is not drawn to scale, right? This prokaryote here on the left would be as big as one of these chloroplasts here, or as big as a mitochondria. And we talked about the endosymbiotic hypothesis. 3 domains. We know the 3 domains, right? 2 of them have something in common. Which 2 have something in common? Bacteria and what? Archaea. They are both what kind of cells? Prokaryotic cells. They are both prokaryotic cells. What's weird about archaea?
It's newer, but
it's newer. They thought it was ancient, hence the misnamed. But they are extremophiles, yeah. And they have little bit differences in their cell membranes and their cell walls in order to enable them to be extremophiles. Good. All right. Good. Phylogeny. So we know, and this would be like a cladogram not leaning, right? So you have a common ancestor here. This common ancestor was the ancestor to A, B, and C. But this common ancestor here was only to B and C. We know that. Okay. So when we look at systematics, that's the study of the diversity of all the organisms. But when we talk about phylogeny, we're looking, if you're looking at the phylogenetic history of it, you're looking for its evolutionary history. So you can work from bottom up or top down in that process. When we talk about cladograms, a clade is a group that includes a common ancestor and all of its descendants. This right here is not a clade. Why? Yeah, they do not share a recent common ancestor. Yes, you could say this whole thing is a clade, right? And this is your ancestor. But if you just highlight this, you would not call that a clade because they're coming from 2 different ones. This is not a clade because you're leaving this guy out, right? But if you pick this or all of these, right, those would be cladograms. Remind you of that. And then what we end up doing is you just keep dropping people off that don't have. At this point right here, everybody's in the club. But you know what? We got to drop off hagfish, agnatha. He's a jawless fish, but everybody else from here on up has jaws. We have some problems here, right? We have problems with the birds, right? Because they're misappropriated. Okay. We already know that. Oh, yes. You want to go with wherever you have the least exceptions is where you want to go. That's Occam's razor, which says, look to see what makes the most sense and probably go with that one. Don't assume some trait is going to de-evolve and then later re-evolve somewhere else. Do you understand what I'm saying by that? Don't set it up that way. Oh, and here it is, parsimony. I just said that. Okay. Homologous structures, we talked about that already. We talked about divergent evolution. You have one ancestor, right, a common ancestor, and you end up from that forming two different species. Convergent evolution, you end up in the same place. You do not probably have a common ancestor, but you ended up because you're dealing with your environment in the same way. So it doesn't matter if you're a bird or a bee, you're both flying. That does not mean you're related, just means you're responding to your environment in the same way. And then parallel example, like I mentioned this earlier, about mammals we have here in the United States and in Australia. We have a mammalian ancestor, but we are both, and we have some similarities. There's like flying squirrels in both, but one is a marsupial and one is a placental. Yay, classification. Domain, then what? Kingdom, phylum, class, order, family, genus, species. Say that with me again. Domain, kingdom, phylum, class, order, family, genus, species. Did King Philip come over for good soup? Sure. Okay, now, as we go in towards soup, are we becoming more or less inclusive? More or less inclusive? Less, because we're becoming more specific, right? There's 3 domains, right? And then, okay. All right, let me skip that. All right, genetic recombination. How can we get variation? We have 3 modes right here. In your lab, we're getting ready to transform. Once we genetically engineer our bacteria, we're going to give it DNA to bacteria that we have created. Conjugation is 2 bacteria having sex. Transduction is when a virus acts as a vector to take DNA from one organism to another. Either way, we can have changes to our DNA, right? And what's another way you could have changes to your DNA that is not represented in this picture? Mutations. Good. All right. Yes, you did it. That is a great place. We got through, I think, some. How long is the next big idea? Let me see. Do you want to try it? Want to do the questions? Sure. Okay, we'll do the questions.
30 seconds.
We have 30 seconds. Oh, if that's it, we're probably going to have to save the questions for next time, and we'll start there. Okay. All right. I'm proud of you. Good job. Are you glad you came?
Yes.
Okay, so it's fast-paced and furious, and I'm going to put on the board that we are on big idea 1C. Good job. I'll see you next time.
Thank you.
You're welcome. Hang in there. Have a piece of toast.